Warm greetings. I am Mrs. J. Albinovins from Holy Cross College, Nagarkoil, presenting a paper on evaluation of bacterial contamination in borehole water with reference to septic tanks. Introduction. India is a country which depends upon groundwater for their utilities. The rapid population growth and increasing industrial activities leads a tremendous increase in water pollution. This is considered as one of the primary reasons for environmental pollution. Water scarcity as well as the water quality are the two important issues of the world that is concerned mainly with global population growth, industrialization and also urbanization. Groundwater is an important source of water supply throughout the world. The activities of human beings is also one of the main causes that leads to the deterioration of surface water as well as groundwater. According to the World Health Organization, the pathogenic microorganisms should not be found in drinking water resources. Water contamination will lead to many waterborne diseases. Most of the contamination arises from the feces of warm-blooded animals as well as the human beings. The sewage generated from home is one of the important issues to make water quality issues. The sewage wastes, both solid as well as liquid, were usually stored in septic tank. These wastewater will migrate and make a proper link with groundwater sources and cause contamination. The contamination is mainly because of microorganisms. Some chemicals like nitrates, chlorides and sulfates also pollute the groundwater. Some of the most common methods used to detect coliform in water includes membrane filter, the most probable number method and also the multiple tube fermentation method. Next is the objective of the present study. The main focus of the present investigation is to identify the presence of microbial contaminants in the groundwater which occurs due to the nearby availability of septic tank. Then methodology. 10 borewell water samples were collected from the village areas of Ilandavilai, Mekod, Murasangod and Palayam in which the septic tanks should be within 0 to 35 meter. A control borewell was also selected in which the radius of the well and septic tank is above 35 meter. After collection, the samples were kept in sterilized bottles and transported immediately to the laboratory for further analysis. Usually, this sampling was done between the hours of 5:30 am and 8:30 am. Coming to multiple tube fermentation test. The multiple tube fermentation technique was performed using chromocult coliform agar. This agar was used to determine the presence or absence of coliform bacteria. In this technique, the borewell water sample was passed through the membrane. This membrane will retain the bacterial isolates present in the sample. After filtration, the membrane with bacterial isolate is placed on the media and then incubated at 37 degrees centigrade for 24 hours. After incubation time, the colonies with salmon to red color were recorded as coliform, dark blue to violet color was recorded as E. coli and the colonies with green to turquoise are considered as salmonella. Total coliforms are recorded when salmon to red, dark blue to violet and turquoise colonies are seen. In this technique, 1 ml of borewell water was added to 99 ml of dilution water. 3 serial dilutions were made with the 99 ml dilution water and 1 ml of resulting solutions. The final solution was filtered through a sterile micropore filter by suction, thereby capturing any coliform bacteria. The chromocult coliform agar was prepared and sterilized and added in the petri dish. Then the membrane was placed aseptically into the agar using sterile forceps. The petri dish was incubated at 37 degrees centigrade for 24 hours and after incubation period the number of colonies with different morphology and pigment formation was noted. Results. This table clearly depicts the total bacterial contaminants present in the sample of borewell water, that is the 10 samples. The sampling results were expressed as colony forming units per 100 which denotes in 100 ml of water. Based upon the microscopic, macroscopic and biochemical characterization, the organisms were also identified. According to the results, the non-fecal coliforms are the most common and prevalent bacterial contaminants found in maximum number in the groundwater sources. The non-fecal coliforms were found in all the borehole samples analyzed. This is because of the availability of septic tank with closeness to the sampling site. So due to this contamination, the borehole water will not be suitable for domestic as well as other purposes. Salmonella, it is also found in the sampling water but with very less percentage, that is 5% only. The presence of Salmonella species also indicates a vulnerable contamination. The presence of E. coli is one of the most specific indicators of fecal pollution. The WHO guidelines recommended that there should be zero colony forming units for E. coli in any 100 ml of water for drinking purposes. In the present borewell sample, the count of E. coli is extremely low, that is 15%, and is completely absent in many sites of sampling. This low percentage is because of the unacceptable distance for septic tanks from groundwater resources. If the enumeration for the total coliform in a sample is high, and the enumeration of fecal coliforms are low or zero, it should not indicate the presence of fecal contamination. It may be due to the entry of soil or organic matter into the water or by the growth of other types of coliforms. The control site sample clearly shows the absence of bacteria. Coming into the conclusion, the present study indicates the presence of microbial contamination with low percentage in all the sites of sampling. It is necessary to perform water quality analysis on all the boreholes in the village at least twice every year. It is also necessary to monitor whether the sewage from septic tanks and other sources enters into different groundwater sources. Effective sewage treatment system must be strictly followed to minimize the groundwater pollution. Proper awareness among the people about the disposal methods of wastes and information about septic tank contamination is also necessary. Thank you all.